Congratulations! You finished up mitosis for lesson 2 in our unit 2 notes. We're going to start lesson 3 today. Now, a little bit of warning. There's going to be a lot of similarities between mitosis and what we'll be learning today, meiosis. We also, though, will discuss the specific differences at the end, but also the similarities. You're going to take advantage of that because if you learned mitosis really, really well, it is going to help us a little bit with making sure that we understand with meiosis and also the differences between those two processes. So have your notes out, get ready to write, and let's get started. So Unit 2, Lesson 3 guided notes, meiosis. Now, meiosis is a form of sexual reproduction. Any organism that is able to do sexual reproduction does this process with their cells. And then they do this process specifically takes place in whatever reproductive organs they have in their bodies. And meiosis is, and you're going to write this down, a form of cell division to produce sex cells, also known as gametes. And these cells have half the number of chromosomes. They're also known as haploid. We'll talk about those terms towards the tail end of our notes. In other words, we take the normal diploid cells that are made in mitosis and whatnot, and we split them further in half to make these half cells, haploid cells. And that, unlike mitosis where there's just one PMAT, meiosis actually has two divisions, and we call that meiosis 1 and meiosis 2. And you can see in the image, we have prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. It follows the PMAT, but it just repeats twice. If it has a Roman numeral 1 or just the number 1, it goes with meiosis 1. If it has the Roman numeral 2 or just the number 2 after it, it goes with meiosis 2. Now, interphase. Interphase is pretty similar to the interphase that we had in mitosis. And chromosomes, they're replicated in the S phase, just like in mitosis. And each duplicated chromosome or replicated chromosome consists of two identical sister chromatids attached at the centromere. You're going to write these things down in your notes. Now, prophase 1. Again, anything that's written on this slide, besides the stuff that's written in the pictures, you're going to write down in your notes. Prophase 1. Chromosomes condense, and the other 3 prophase things. So remember that chromosomes condense, we have spindles form, the nucleus disappears, the centrioles move to the poles. Those 4 things that happened in prophase and mitosis, they happen here, but we're not going to take our time to write those because there's so much other stuff that we've got to know about prophase 1. So chromosomes condense and the other 3. Then we have homologous chromosomes form a tetrad. So homologous means same. The same chromosomes come together to form a tetrad, or 2 chromosomes making 4 chromatids together. So you'll notice that they pair up in our pictures. And this part is the very first part of genetic variation that occurs. Remember, in mitosis, we were making identical cells. We do not make identical cells here. And that's because something happens. It's called crossing over. And those tetrads cross over the sort of arms of the chromatids, and they actually switch information. And again, we'll talk more about crossing over later. Metaphase 1. Metaphase 1, tetrads align in the middle. Again, remember, metaphase, middle. So pretty much same thing. They line up in the middle, but one more additional thing happens just in metaphase 1. A process called independent assortment occurs here. And it's random and it causes genetic variation. And what I want you to look down at the picture that's sort of down at the bottom again, that's the 4 blue cells, you'll notice that those are options. Look at the location of the red and blue, and you'll notice that each circle represents a cell, has a different combination of what's on what side. And again, it's random. So that provides more genetic variation. And again, we'll talk more about independent assortment later. Anaphase 1. Homologous chromosomes or tetrads separate or pulled apart. So remember, anaphase apart or anaphase away. So they're pulled apart here too. Now, what we want to pay attention to and what I want you to underline once you write it down is that this part, this is the last point where we will hear homologous chromosomes or tetrads. If you hear homologous chromosomes or tetrads, you know that you were talking about prophase 1, metaphase 1, or anaphase 1. If we hear sister chromatids, we're talking onward in the process, and that's going to help us figure out what's the difference between anaphase 1 and anaphase 2. So sister chromatids now are together. Okay, telophase 1, the very last phase of meiosis 1. Each side now has a haploid set of non-identical chromosomes. Remember, we're no longer dealing with identical because of crossing over independent assortment. So we're now dealing with unique, non-identical. Cytokinesis is going to occur towards the tail end of this. You'll see that you have 2 nuclei forming, splitting of cells starting. All the normal telophase stuff is happening, but we just need to know that now we have 2 nuclei, and we're dealing with a non-identical and a unique cell. Those are our key terms. So 2 cells forming, 2 nuclei, but we're dealing with unique, not identical. And at the very bottom, you'll see on your next page of your notes at the very top, you're going to write this sentence about cytokinesis, where the cell splits into 2 unique, non-identical haploid cells. And they're haploid cells because it really doesn't have the information. It's got half the amount of information that needs to be utilized. Now we're going to jump to prophase 2. Prophase 2, the same 4 things that happen in prophase and mitosis happen here. DNA condenses, nucleus disappears, spindles form, centrioles move to the poles. The only visible difference that you're going to be able to tell is that instead of seeing just one cell do this, anytime you see 2 cells that look like they're in prophase, we know we're in prophase 2. So the normal prophase things, except we're dealing with 2 cells rather than just 1. So 2 cells are having all this happen. And again, you'll see a picture of 2 cells. Remember that these pictures, these images that are in your notes are going to be the same images that you'll see during your quizzes and tests. Metaphase 2, same thing. Again, chromosomes line up in the middle. It's really what we need to know. Spindle attaches to the center of the chromosomes. That happens in all metaphases. But the main thing is we have chromosomes lining up in the middle, and we see 2 cells doing this process. And you'll notice I'm no longer using the words homologous chromosomes or tetrads. Anaphase 2. Again, anaphase apart. Copied chromosomes move apart or pulled apart to the ends of the poles. You might also hear this is where the sister chromatids separate. And again, difference being you know it's in meiosis 2 somewhere because you see 2 cells doing this process at the time. Telophase 2. Nuclei reform. We get 4 nuclei. Chromosomes are going to uncoil, spindle disappears. The normal things except for instead of having 2 nuclei forming, remember telophase 2, we get 4. And these 4 cells are 4 haploid unique daughter cells, or also gametes. In females, these gametes are called eggs. In males, these gametes are called sperm. And this is also cytokinesis occurs at the end of this. So if you see nuclei reforming, you start to see distinct cells, and you see 4 cells. You see 4 cells ever. Your hint is telophase 2. You see 4 cells, telophase 2, always. And of course, you're going to add to the very bottom of your notes. Cytokinesis occurs here. Both cells split into 2, forming 4 haploid, unique, non-identical cells. So at the end of meiosis 1, we had 2 haploid, unique, non-identical cells. At the end of meiosis 1, we had 2 unique ones. At the end of meiosis 2, we have 4 unique cells. Now, when I said that we will revisit crossing over an independent assortment, this is where we're going to have. You'll have some questions about, where does genetic variation come from? You do not look the same as your siblings. You're not all twins. Why? Because of these processes. These processes make you unique. We'll talk later about how twins occur, both fraternal and identical, but this is why you look similar to your siblings, but you do not look the same. Because you're made up of half of your mom and half of your dad. And that even those halves are jumbles of the complete part of what makes up your mom and dad. So crossing over happens in prophase 1. This is where the tetrads exchange alleles. So they sort of switch little small pieces. At this point onward, you're not creating identical cells anymore like mitosis. It's completely unique. So this is the first part. It happens in prophase 1. The second part of genetic variation occurs in independent assortment, which happens in the next phase, the next stage, metaphase 1. And this is where, yes, the chromosomes, the tetrads line up in the middle, but depending on whether one lines up on the right or the left, as you can sort of see, will create a variety of differences. So 2 types of genetic variation occur. One in prophase 1, one in metaphase 1. After that, cell's completely unique. Now, diploid versus haploid, we've used those terms before. Diploid is what the end result of a cell in mitosis is, just regular, doing its business, 2 complete sets of chromosomes. In meiosis, we take it a step further and we keep having them, having haploid. So we're creating haploid cells, which contain only 1 set of chromosomes, and it's really only half of what an organism needs to survive. And that's okay, because we take 1/2 in an egg, 1/2 in a sperm, we fuse them together during fertilization, and we get 1 complete set. Now, here's your overall picture. You'll notice that these images are the same images throughout your notes, and also will be the same images that are in your quizzes and tests. Now, unlike normally where you have to do the practice by yourself, today I'm going to do the practice with you. That's why this video is a little bit on the longer side. We're going to be comparing our knowledge about mitosis to our knowledge about meiosis. That's going to help us be able to differentiate and also use the similarities to our advantage. So let's talk about mitosis first. We're going to copy this down into our notes. The purpose of mitosis that makes body cells are also known as somatic cells. It's for growth, repair, replacement, and it's just dividing the nucleus. That's its purpose. It starts with 1 diploid cell. It ends with 2 identical diploid body cells. And you will need to know that that's associated with mitosis. How many stages? There's 4, just PMAT. How many division? There's only 1 cytokinesis here, just 1, at the end of telophase. Now with meiosis, we have a couple things that both meiosis 1 and meiosis 2, just in general, we'll just summarize. First thing is that meiosis makes sex cells. That's the purpose of it. It starts with 1 diploid cell, just the same as mitosis, but this one ends with 4 unique haploid cells, or 4 unique gamete cells, or sex cells. This one has 2 divisions, one at the end of meiosis 1 and one at the end of meiosis 2. Now in meiosis 1, again, we have P1, M1, A1, T1. PMAT, just with 1s at the end of them. Starts with one diploid cell, ends with two haploid unique cells. Crossing over occurs in prophase 1. Independent occurs in metaphase 1. Those are the big differences, what we end with and those two genetic variations that occur, because they don't occur anyplace else. In meiosis, we have our P2, M2, A2, and T2 to stand for our prophase, metaphase, anaphase, telophase. And again, the 2 is at the end. That tells us it's in meiosis 2. We're going to be seeing 2 cells at this point. We'll start with 2 haploid cells. We'll end with 4 haploid cells, and these are unique. And honestly, there's no genetic variation occurring here, so there's less occurring. Now make sure that you have all of this written in your notes. And take a last look at this last picture. With humans, we have 46 chromosomes. Our normal cells are diploid. Remember that we had to double them at interphase to 92, then they get divided first to get to 46. And then at the end, because we had started with the 92, when they split, we get 46 and 46. That's diploid. In meiosis, we start out in interphase with 92. We start the process of splitting during meiosis 1. We split again, and each of those cells end up with 23 each. And if a human cell has 23 chromosomes, that is a haploid or sex cell. Now this completes our notes. I want you to have a wonderful day, and remember, this is a bit tricky, so make sure you come to the review sessions. Have a great day.